Hi friends, my topic is somaclonal and gametoclonal variations. The genetic variations that we can see in vitro cultured cells are called somaclonal variations. Such plants are called somaclones. Cultures obtained from callus are called caliclones and the cultures obtained from protoplastis are called protoclones. The term somaclonal variations was first used by Larkin and Scowcroft in 1981. The variations that we can see in plants regenerated from gametic cells like anther cultures are called gametoclonal variations. The variations observed in plants obtained from protoplast cultures are called protoclonal variations. Nomenclature. Generally, two symbols are used. Chalef has labeled plants regenerated from tissue culture as R or R0 plants and the self-fertilized progeny of R0 plants as R1. The subsequent generations by self-fertilization are R2, R3, R4, etc. Larkin and Scowcroft have referred regenerated plants as SC1, which is similar to R0, and the subsequent self-fertilized generations as SC2, SC3, SC4, etc. The basis or reasons of somaclonal variations are different, such as karyotype changes, changes in chromosomal structure, single gene mutations, mitotic crossing over, gene amplification and nuclear changes, transposable elements, and DNA methylation. We can check all these individually. First one is karyotype changes. The most frequently observed chromosome abnormality is polyploidy. So what is polyploidy? It is the presence of more than single pair of each chromosome. In oats, potato, barley, we can see aneuploidy, which is the loss or gain of individual chromosome from normal chromosomal set. The change in chromosome number in a variant plant is maybe due to reduced fertility or due to altered genetic ratios in the progeny of self-fertilized plants. Chromosome structural changes. We can see translocations in potato, ryegrass, oats, etc. And also chromosome deletions, duplications, inversions in regenerated plants. Example, ryegrass or barley. Chromosomal irregularities such as breaks, eccentric is the lack of centromere, and centric fragments, ring chromosomes, micronuclei were also observed in garlic somaclones. Here the ring chromosomes is the chromosomes whose ends are fused to form a ring. Cytological abnormalities include multi-lobed nuclei, multinucleate cells, abnormal anaphase, and mixoploidy. Mixoploidy is the there occurs two cell lines, that is one diploid and one polyploid, which coexist within the same organism. Single gene mutations. The regenerated plants and their progeny may show phenotypic changes, and these phenotypic changes may be due to single gene mutations and would be inherited in a Mendelian fashion and show recessive traits. Some of the single gene mutants are chlorophyll deficiency, dwarfs, seed traits, reproductive structures, and necrotic leaves, and these are observed in maize. Mitotic crossing over. Mitotic crossing over can occur as symmetric and asymmetric recombination. It accounts for recovery of homozygous recessive single gene mutations in some regenerated plants. If asymmetric recombination occurs in sister chromatid exchange in somatic cell, then it may lead to deletion or duplication of genetic material and may cause variation. Gene amplification and nuclear changes. Nuclear genes are affected by tissue culture stress. Both AT and GC rich fractions are amplified in tissue culture derived plants. Plant cells can increase or decrease the quantity of a specific gene product by differential gene amplification and diminution. Diminution is the reduction in size. Reduction in ribosomal RNA genes also found in potato plants regenerated from protoplastos. Transposable elements. The activation of transposable elements also cause variation. Heterozygous light green somaclones with a high frequency of colored spots on the leaf surface is detected for a clone of an tabacum. Genomic changes in a maize line and mutations in tobacco line due to activation of transposable elements under in vitro tissue culture condition is also observed. DNA methylation. In some sites of the tissues, they are methylated and here the gene is not expressed. But in some other tissues, the sites are non-methylated and the gene is active. Thus, we can say that an active gene is described as unmethylated. It can also enhance quantitative trait variation as several genes can be affected simultaneously. The transcription of the gene is disabled by methylation and also it regulates the gene expression during somatic embryogenesis. The somaclonal variants can be isolated by two methods, without in vitro selection and with in vitro selection. Without in vitro selection, here we take an explant and is cultured in a medium. We supply the growth regulators, but in this method we do not introduce selective agents. These cultures are then subcultured normally and transferred to shoot induction medium for regeneration. The produced plants are grown in pots, transferred to field and analyzed for somaclonal variants. Here, as you can see, the explant, here we have taken a leaf and from the explant callus is formed and from the callus shoot regeneration occurs. From this shoot regeneration a plantlet is developed which is transferred to the field and screened for the desired traits. After screening, the somaclonal variants are selected and taken for agronomic trials. By this method, we have developed many somaclonal variant crops such as sugarcane, potato, tomato, cereals, etc. Here we can see it is a table of selected list of disease resistant croplands obtained from somaclonal variations without in vitro selection along with the pathogenic organisms. Limitations of without in vitro selection approach. The main limitation is that no particular directed or specific approach is there for the isolation of somaclones. The appearance of a desired trait is purely by chance and this process is time consuming and requires a screening of many plants. With the in vitro selection, it involves handling of plant cells in cultures like microorganisms and selection of biochemical mutants. The main difference with in vitro selection is that we introduce a selective medium in the culture. The cell lines are screened from the plant cultures for their ability to survive in the presence of this selective medium, which can be a toxic or inhibitory substance or under conditions of environmental stress. As I said earlier, the differentiated callus which is obtained from an explant is exposed to inhibitors or the selective medium which can be a toxin, antibiotic or amino acid analog. Several selection cycle is carried out to isolate the callus cultures which are tolerant and these calli are then regenerated into plantlets. These plants so obtained are in vitro screened against the toxin or pathogen or any other inhibitor. The plants which are resistant to the toxin are selected and again further grown by vegetative propagation or self-pollination. Further, the subsequent generations are analyzed for the resistance to this particular disease against this specific pathogenic organism. And not only disease resistant plants, we can also develop plants which are resistant to herbicide, antibiotic, etc. using this approach. Here we can see this is a diagrammatic representation of the process with the in vitro selection. First of all, we select a plant, and from the plant we isolate the explant and from which the callus is formed. The callus is maintained and proliferated. At the same time, we also need the selective medium or the toxin or the inhibitory agent. For that, we select a particular pathogenic organism and is cultured. And this culture filtrate is purified, and then the toxin is isolated from this culture filtrate. Toxicity determination is done for lethal concentration, and this callus as well as the isolated toxin is introduced into the same medium. Several selection cycles occurs and the tolerant calli is isolated from this and undergoes regeneration into a plantlet. In vivo screening against the toxin or pathogen occurs, and by self-pollination or vegetative propagation, the progeny clones are obtained from the each plant. Then test for disease resistance is done, and the such resistant plants are taken for generation and then used for agronomic trials. Advantages of with the in vitro selection approach. The major advantage is that we can select for desired trait rather than a general variation. The procedure is less time consuming when compared to without a in vitro selection approach. Here in this table we can see some of the selected examples of disease resistant crop plants obtained by in vitro selection. Here we can also see the selection agents used, and here the CF represents culture filtrate and CET represents crude toxin. Factors affecting the production of somaclonal variants. There are different factors. First one is genotype and explant source. The nature of genotype of the plants influences the frequency of regeneration and frequency of production of somaclones. Explant can be taken from any part of the plant such as leaves, roots, internodes, ovaries, etc. But also the source of explant is critical. For example, potato plants regenerated from callus of rashes and petiole are much higher when compared to those regenerated from callus of leaves. Cell culture duration. For many plant cultures, somaclonal variations are higher with an increased duration of cultures. That, for example, genetic variability in tobacco protoplasts were increased from 1.5 to 6% by doubling the duration of cultures. Growth hormone effects. We use plant growth regulators in the medium, and this may influence karyotypic alterations in cultured cells and therefore development of somaclones. Growth hormones such as 2,4-D and naphthalene acetic acid are used to achieve chromosomal variability. Selection of somaclones with in vitro selection are dependent on many parameters. First of all, selection propagule. Any tissue can be selected, such as protoplast or calli, and each have their own disadvantages as well as advantages. But our ultimate aim is to get a high frequency regeneration from that particular tissue. Selection from calli is probably the least complex method. Callus culture is easy to perform, and the maintenance and handling can also be done without much difficulty. Next is selection agent. As we have seen earlier, with the in vitro culture, mainly we added the selection agent. Here the inhibitor can be amino acid analog, herbicide, or a synthetic toxin isolated from fungal liquid culture. For salt and aluminum tolerance, specific compounds such as sodium chloride for salt and aluminum chloride for aluminum tolerance is used. The inhibitor concentration used in the selection experiments is depend on the sensitivity of the plant cell line. To select a disease-resistant mutant, we use toxins or crude toxin or purified culture filtrate as a selection agent. So, to select that, first of all, we should check the toxicity of these toxins, and only then the concentration of the toxin to be used will be known. To check the toxicity of selection medium or the toxins, the leaf or root bioassays are carried out. Then a particular concentration of the toxin is used for the selection of calli cells or protoblasts. Next is selection technique. The cells is incubated in an inhibitor containing medium for a particular time or sufficient length of time to ensure the expression or growth of the resistant clones. The selection can be carried out in different ways using different cycles, different number of cycles, such as like in very high toxin or inhibitor concentration with many or just one cycle of selection, or many cycles with a pause, or non-toxic media after second or third selection cycle, or on an increasingly higher inhibitor or toxin level. The selection technique involves placing pieces of cultured tissue on a medium containing a near lethal concentration of inhibitor and selecting the fast growing visible sectors of tissue from the remainder of the cultured material. Resistant tissue obtained in this way continues to proliferate on a medium containing high level of inhibitor concentration. Next is the ability for regeneration of plants. Usually, when the tissue culture is exposed to continuous subculture, their capacity to regenerate the plants is lost over a long period. Regeneration is generally reachieved by reducing the level of oxygen in the media and incubating the cultures in light. Next is in vivo testing. To make sure that the plants developed are resistant towards that particular pathogen, first testing against the toxin of pathogen, then they are tested against that pathogen itself. Conditions required for the development of disease symptoms in plants should be provided, and thus they are tested. Resistance stability. One of the major problem faced is that under selective pressure, the plant cell lines show resistance to a particular inhibitor or toxin. But when removed from this selective pressure, they show characteristic instability. In order to test the stability of the resistant trait, at least two cultures for each selected line is established. One is subcultured routinely in the presence and one in the absence of selective conditions. At various intervals over a period of several months, perform dose response test to maintain the resistance stability. Further, the R0 plants which were found to be resistant should be analyzed for resistance to that character in R1 and in the subsequent generations. Applications of somaclonal variations. Production of agronomically useful plants. Due to somaclonal variations, several novel variants of existing crops have been developed. For example, pure thornless blackberries and somaclonal variations with useful and improved morphological characters are also obtained. For example, rice, wheat, maize, sugarcane, potato, carrot, etc. Resistance to disease. They largely contribute towards the development of disease resistance in many crops. For example, rice, wheat, maize, sugarcane, tobacco, apple, tomato, etc. Resistance to abiotic stresses. It is possible to develop biochemical mutants with abiotic stress resistance. For freezing tolerance, example wheat, salt tolerance, rice, maize, tobacco, aluminium tolerance, example carrot, sorghum, tomato, etc. Resistance to herbicides. The selected examples are tobacco resistant to glyphosate, sulfonyl urea and picloram. Carrot which is resistant to glyphosate. Lotus which is resistant to 2,4-dichlorophenoxyacetic acid is obtained. Improved seed quality. New variety of Lathyrus sativa seeds with a low content of neurotoxin is developed. The neurotoxin is beta N-oxalyl 2-alpha beta diaminopropionic acid or ODAP. This neurotoxin indicates the potential of somaclonal variations for the development of varieties with improved seed quality. Insect resistance. Cali from susceptible wheat cultivar Stevens were exposed to an extract from aphid, and it showed resistance to aphid in both the R2 and R3 generations. Variant of Bermuda grass called Brazos R3 or Cynodon dactylon with increased resistance to fall armyworms were observed. Limitations or the disadvantages of somaclonal variations. The first one is that somaclonal variations may not always be useful. The variations occur in an unpredictable and uncontrolled manner. The genetic traits obtained by somaclonal variations may not be stable or heritable. Since somaclonal variations are cultivar dependent, it is a time consuming process. Somaclones can be produced in only those species which regenerate to complete plants, and also many cell lines like callus may not exhibit regeneration potential. Gametoclonal variations. The variations observed in the gametic cells are regarded as gametoclonal variations, unlike the somaclonal variations, which are the variations detected in cultures of somatic cells. As the somatic cells divide by mitosis, the genetic material is equally distributed to the daughter cells. In contrast, the gametes, being the products of meiosis, possess only half of the parent cell genetic material. Difference between a gametoclonal variation and somaclonal variation. Mainly three differences. First one is that mutants obtained from gametoclonal variants give rise to haploid plants since a single set of chromosomes are present. In gametoclonal variations, the meiotic crossing over is the recombination process. The gametoclones can be stabilized by doubling the chromosome number. The production of gametoclones, they can be developed by culturing male or female gametic cells. Cultures of anthers or isolated microspores are widely used. Improvements is made in several plant species through development of gametoclones. For example, rice, wheat and tobacco. Source of gametoclonal variations. Cell culture technique induce genetic variation. Variations may be induced while doubling the haploid chromosomes. Genetic variations may occur due to heterozygosity of the diploid. So that's all about somaclonal variations and gametoclonal variations. Thank you.